In the last video lecture we understood what are Taggart-labeled immunoassays. We also discussed two important tags, radioisotopes and enzymes. The technique in which a radioisotope is used as a Taggart label, that is, radioisotope is covalently linked to antigen or antibody for the detection of antigen-antibody complex, is known as radioimmunoassay. Radioimmunoassay was developed in 1959 by endocrinologist Rosalind Yalow and Solomon Burson to measure the circulating levels of important hormone insulin. In 1977, Rosalind Yalow was awarded Nobel Prize for Medicine for the development of radioimmunoassay for peptide hormones. Solomon Burson could not share the award because of his untimely death in 1972. In radioimmunoassay, the antigens, or antibodies, are labeled with radioisotopes. We know that radioisotopes emit radiations. These radiations can be detected and measured using specialized instruments such as gamma counter. Most popular radioisotope used as a tag is iodine-125. Let's first understand the basic principle of this assay. Radioimmunoassay determines the concentration of an antigen in a sample based on the competitive binding between radiolabeled and unlabeled antigen for its specific high-affinity antibody. Suppose this is a microtiter well. A fixed or constant amount of antibodies specific to our test antigen is immobilized in this well, say, 2 antibodies. Next, we add specific radio-labeled antigens to this well. And the amount of these antigens is such that they saturate all the antigen binding sites present in the well. This well is rinsed to remove the unbound radio-labeled antigens. At this point, if we measure the radioactivity of the well, it will be maximum, say 100%. Now in a second microtiter well, again we take the same amount of immobilized antibody. But this time, we add unlabeled antigens along with the previous fixed amount of radio-labeled antigen. Note that antibodies present in the well has same affinity for both the labeled and unlabeled antigen, since both the antigens are same. Therefore, these antigens will compete with each other for the antigen binding sites. And now, some of the antigen binding sites will be occupied by the unlabeled antigen. Again, after binding takes place, the well is rinsed to remove any unbound antigens. If we now measure the radioactivity of this well, it will not be 100%. It will decrease because the unlabeled antigens have replaced some of the radio-labeled antigens. Decrease in radioactivity tells us that the antigen is present in the test sample. But how to determine the concentration of this antigen? Suppose we want to detect presence and quantity of an antigen in the patient's serum. We suspect that this patient's serum has the antigen of interest. To quantitate the antigen present in the patient's serum, first we need to obtain a standard curve. This standard curve is obtained by measuring the amount of radioactivity using fixed and excess concentration of radio-labeled antigen and varying known concentrations of the unlabeled antigen. Again, note that both these radio-labeled and labeled antigens are same as the antigen we are going to detect and quantitate in the patient's serum. Let's have a look at the basic steps involved in the generation of standard curve. We require 3 main components. An important point is that all these components and their quantity is known to us. First component is specific antibody to the test antigen. The quantity of this antibody being used is fixed. These antibodies are immobilized in microtiter wells. Second component is the radio-labeled antigen. Asterisk symbol used in this designation tells that the antigen is labeled. The quantity of radio-labeled antigen is again fixed. This quantity is enough to saturate all the antigen binding sites in the well. Third component is the unlabeled antigen. We will use increasing varying concentrations of unlabeled antigen. First, we take a microtiter plate. To keep this illustration simple, we are showing only 5 microtiter wells. In each well we will immobilize fixed amount of antibodies specific to the test antigen. Let's say 3 antibodies are fixed in each microtiter well. Thus, concentration of antibody is constant in all these wells. Now, we add fixed amount of radio-labeled antigen to the first well. This amount of radio-labeled antigen is enough to saturate all the antigen binding sites in the microtiter well. We will rinse the well to remove any unbound antigens. If we measure the radioactivity of this well, it will be maximum. Let's say 100%. This is our control well. Next, we will add same fixed amount of radio-labeled antigen in all other wells too. But along with this, we will add unlabeled antigen in increasing varying concentrations. That means, to each well, fixed amount of radio-labeled antigen and increasing amount of unlabeled antigen is added. What will happen now? As we said before, antibodies present in the wells cannot distinguish between radio-labeled and unlabeled antigens. Thus, in each case, the unlabeled antigen will compete with the radio-labeled antigen for the antigen binding sites. It is important to note that, as the concentration of unlabeled antigen increases, it occupies more antigen binding sites. Now, in each well there will be bound as well as unbound antigens. In the next step, we will separate the unbound antigens by rinsing the wells and measure the radioactivity in each well by gamma counter. Radioactivity will decrease in each well with increasing amount of unlabeled antigen. This is because now some of the antigen binding sites have been occupied by unlabeled antigens, and there is less number of bound radio-labeled antigens in each of these wells as compared to the control well. At the end of this procedure, we have value of radioactivity for each well. Also, we know the concentration of the unlabeled antigen added in each well. Let's now plot a standard curve. In this graph, y-axis represents the radioactivity measured for each well, and x-axis represents the concentration of the unlabeled antigen. As the concentration of unlabeled antigen increases, the value of radioactivity decreases. So, this is our standard curve. Note that we are showing only the linear portion of the curve. Let's now quantitate the antigen in the patient's serum. We will take same quantity of immobilized antibodies in a microtiter well as we used in the standard curve. To this well, we will add a fixed amount of radio-labeled antigen. Again, this amount is same as we used in standard curve. And along with this, we will add suspension of patient's serum. The antigens present in the patient's serum will compete with radio-labeled antigens for antigen binding sites in the well. Well is rinsed to remove unbound antigens, and radioactivity is measured. Let's suppose radioactivity measured for this well is 35%. Decrease in radioactivity indicates the presence of antigen in the patient's serum. We will extrapolate this value of radioactivity in the standard curve. Thus, we will get to know the quantity of antigen present in the patient's serum. Radioimmunoassay is mainly used to detect hormones and to diagnose allergies. These tests are now replaced by more sensitive and cost-effective methods. The main disadvantage of using radioimmunoassay is the hazard to the professional due to exposure to radioactivity. Most of the radioisotopes have short shelf life. This technique is also expensive as it requires specialized equipment. Moreover, it is a time-consuming technique. That's all in today's video lecture. In next video we will study enzyme-linked immunosorbent assay. Thank you for watching. ELISA stands for enzyme-linked immunosorbent assay. ELISA is a labeled immunoassay where the antigen or antibody is labeled with a reporter enzyme. Recall that reporter enzymes produce a color change when the enzyme reacts with its specific substrate. Thus in ELISA, color change is used to identify a substance. This assay is used for the detection and quantitation of substances such as peptides, proteins, antibodies, and hormones. There are three main formats of ELISA. These are direct ELISA, indirect ELISA, and sandwich ELISA. In this video lecture we will study direct ELISA. In the previous video lecture, we understood the difference between the term direct and indirect detection assay. Easiest way to remember the difference between direct and indirect ELISA is to look at the enzyme-linked antibody. If enzyme-linked antibody is directly binding the antigen, it is direct ELISA. And if enzyme-linked antibody is not directly binding to the antigen, then it is indirect ELISA. As you can see here, in indirect ELISA, the enzyme-linked antibody binds to the antibody already bound to the antigen. Let's now understand the steps involved in direct ELISA. Direct ELISA is used to detect antigens in the test samples. Test result can be positive or negative depending on the presence and absence of the antigen in the test sample. Suppose we have serum samples from two patients, patient 1 and patient 2. For our illustration, we assume that antigen is present in patient 1 and absent in patient 2. Let's see how to detect and confirm this by ELISA test. We will take two microtiter wells, each corresponding to the patient's serum sample. So this is 1 and this is 2. In the first step, the antigens present in the patient's serum are absorbed and immobilized in the microtiter well. No adsorbed antigens are shown in the well 2 because the target antigen is absent in the serum sample of patient 2. Note that we are not showing other antigens present in the serum of patients. Both these microtiter wells are rinsed to remove any unbound antigens present in the well. In the second step, enzyme-linked antibodies specific to the target antigens are added in each well. These enzyme-linked antibodies will bind to their specific antigens in the well, if they are present. Thus, here you can see that antigen-antibody binding takes place in the first well, and no antigen-antibody binding takes place in the second well. This is because there was no target antigen. Each well is rinsed again to remove any unbound antibodies. After rinsing step, only antibodies present in the first well are those bound to the antigen, whereas no antibodies are present in the second well. In the third and final step, we add substrate specific for the enzyme that is linked to the conjugated antibody. After some time, color development can be seen in the first well. This is because the enzyme linked to the antibody acted on the substrate and resulted in the formation of colored end product. But in the second well there is no color change. This is because there was no enzyme-linked antibody present in this well. Thus, in ELISA test, a color change reveals the presence of the antigen. This is termed as a positive test. No color change indicates that the antigen was not present in the test sample. This is termed as a negative test. Thus, direct ELISA detects the presence of antigens in a test sample. It is commonly used for immunohistochemical staining of tissues and cells. That's all in today's lecture. In the next lecture we will study indirect ELISA. Thank you for watching. This is a pregnancy strip having 3 zones of function. Recall that reaction zone is made up of monoclonal, enzyme-conjugated, anti-HCG antibodies. These antibodies are also known as mouse antibodies. And these antibodies are loosely attached to the strip. Test zone is made up of immobilized, polyclonal, anti-HCG antibodies and inactive dye molecules. And the controlled zone is made up of immobilized anti-mouse antibodies and inactive dye molecules. The anti-mouse antibodies are specific for the reaction zone antibodies. Let's first understand working of positive pregnancy test. Urine sample is applied in the sample port of the pregnancy test strip. And here, we assume that the urine applied has been taken from a pregnant woman. Thus, HCG is present in this urine sample. These are HCG molecules. Urine is drawn up the strip by capillary action, and it arrives at the reaction zone. The monoclonal, enzyme-conjugated, anti-HCG antibodies present in this zone recognize and bind to the specific epitope on HCG molecules. As we said before, these antibodies are loosely attached to the strip. So, as urine arrives in this zone, these antibodies get detached and travel along with the urine by capillary action. Here note that HCG-bound enzyme-conjugate antibodies and unbound enzyme-conjugated antibodies are moving towards the test zone along with the urine. Now, let's see what happens in the test zone. Recall that in the test zone, polyclonal anti-HCG antibodies are present. These antibodies bind to the HCG molecules of the HCG enzyme conjugated antibody complexes. Here, you can see that HCG is now sandwiched between the two antibodies. That is, between the antibody of the reaction zone and the antibody of the test zone. Again, as we said before, the antibodies of the test zone are fixed. Therefore, these test zone antibodies and the sandwich complexes formed by them do not move further along with the urine. But other free anti-HCG antibodies of the reaction zone continue to move further in the strip by capillary action. Next, the free or unbound reaction zone antibodies arrive at the controlled zone. We know that reaction zone antibodies are also known as mouse antibodies. In the controlled zone, anti-mouse antibodies are present which recognize and bind Fc region of the mouse antibodies. Now, look at the test and controlled zone. We know that both zones have inactive dye molecules. And now, in both cases enzyme-conjugated antibodies are also present. The enzyme will act on the inactive dye molecule. And on activation, this dye will result in the color development. Thus, both lines, that is T-zone line and C-zone line, will be colored. This is a positive test. In a negative pregnancy test, there is only one colored line, and that is in the C-zone. Let's find out how this happens. Suppose urine sample which is applied in this pregnancy test strip does not have HCG. The urine sample will flow to the reaction zone by capillary action. Now, when urine arrives at the reaction zone, the antibodies of the reaction zone has nothing to bind to. This is because urine lacks HCG. But, because the reaction zone antibodies are loosely attached to the strip, they get detached and move further with urine. When these reaction zone antibodies reach the test zone, they cross this zone without any interaction. This is because test zone antibodies are specific for HCG molecules. But now, no HCG is present here. Now these reaction zone antibodies reach the controlled zone. Recall that controlled zone antibodies recognize and bind the reaction zone antibodies. So, they bind these antibodies and fix them. Now have a look at test zone and controlled zone in a negative pregnancy test. There is no enzyme-conjugated antibodies in the test zone. Therefore, there are no enzymes to act on the inactive dye molecules. Thus, no color development takes place. But in the controlled zone, enzyme-conjugated antibodies are present. So, enzyme acts on the inactive dye molecule and colored line results. Thus, a single colored line, that is, in the controlled zone, indicates that the test is negative.